Sorry about that. 1 second here. Okay, so first of all, I just wanted to say that I feel like maybe my talk doesn't fit in with the previous talks. On one hand, I am about to blast you with some basic science and some really bright colors. But the take-home or the translational aspect of this talk, I believe, is really important. And so just to clarify, I am the only one so far that does not yet have my doctorate. So I'm entering the big leagues now. Or I like to think so. And I'm coming from the University of New England, the College of Osteopathic Medicine, where I am actually a PhD student through the University of Maine, the Graduate School of Biomedical Science and Engineering. And we're working with Dr. Adrian Morin here at MMC Cardiology as part of a collaboration for this project. So without further ado, I'm going to start to tell you, keyword being start, to tell you a little story. Actually, a big story about a little organelle called the primary cilium. And interestingly, and given the theme that was introduced this morning by Dr. Rosen, this organelle was actually discovered over 50 years ago, around the 1950s, and was sort of discounted because maybe it was too small or maybe it was insignificant. Perhaps it was a vestibular structure. And it has, in fact, resurfaced as something that has become very clinically relevant, in particular in the context of renal disease. And now we're starting to recognize it in the context of many laterality defects, and in particular, and what I'm going to be presenting on today, congenital heart defects. So defects in the context of proper alignment of the cardiac outflow tract, as well as septation, so the proper dividing of the heart into the 4 chambers that we hope that it forms in the adult heart, in the healthy adult heart. So the primary cilium is actually a microtubule-based, sorry, I'm a little nervous, I shouldn't point when I do that. My rule is normally don't do that until a couple slides in, otherwise it shakes. So it's a microtubule scaffold, and the cilium is actually quite small. It's around 1 to 3 micrometers in length, and it extends from the plasma membrane of nearly all eukaryotic cell types. It is a structure that really functions like a cellular antenna to interpret the environment around it, but to also play a critical role in the sensory function of the cell. In other words, translating communication from the extracellular as well as the intracellular environment and the subsequent modifications in gene expression. So the protein that we use to target the gene rather that we use to target the cilium in our lab is a gene called IFT88, which encodes for a protein on the axonal protein transport system that goes in both the anterograde and retrograde directions of the cilium. So that's how we can genetically get rid of this cilium, to study what happens as a result of the loss of the cilium. So it's very small, and in terms of the primary signaling pathways, we often think about a receptor called patch1, which is a receptor that in the presence of, or is known to bind to a ligand called hedgehog. I know I just couldn't resist using my little animation there. But how we manipulate this signaling pathway is actually by using or targeting another receptor called smoothened, here abbreviated SMO. And in fact, in the context of using a mutant smoothened receptor, which functions in the absence of binding to hedgehog ligand. And I'll get to explaining that a little bit more later in the talk. But the important message is that, again, the cilium is functioning like a cellular antenna, and it is telling the cells where to go, how to organize themselves, and how to modify gene expression, in this case, during heart development. So this is just one picture to show you how small they are. This is a 40x zoom 3 objective. So each blue dot is a cellular nucleus, and each little tiny green dot there is a primary cilium. So they're very, very small. And in particular, we're studying a progenitor population of cells called neural crest cells, and in particular, neural crest cells that migrate and contribute to the developing heart, where we then call them cardiac neural crest cells. So without going into details about that population, they historically have thought to contribute to both the outflow tract as well as the developing heart valves, and play a role in contributing to the healthy formation of those, but also, and as I'll show later in the talk, our lab has shown evidence that they're also contributing to the ventricular myocardium, which is, for anyone that studies neural crust, a novel and very sort of controversial outcome. So the big research questions to think about as we go through here is to think about, or is to really try to address as directly as possible, do these primary cilia modulate the pathogenesis of congenital heart defects? And furthermore, this is really a question we're still chipping away at, is to ask if this mechanism is really directly resulting from something going on with hedgehog signaling in particular. So the basics of our design, we use a neural crest specific Cree driver, and in fact, I don't have time to go into detail, but we actually have utilized multiple neural crest specific Cree drivers to show this same phenotype that I'll go over. So Wnt1 Cree combined with an IFT88, again, that cilia specific gene, to create or generate a Wnt1 Cree primary cilia conditional knockout. So in other words, we're getting rid of the cilia of cardiac neural crest cells and studying that phenotype, and also trying to address what mechanistic considerations to target next. We're also using a TD tomato reporter, which is just a fluorescent gene that we're using to label the cells that we're interested in. So here they are, those bright, obnoxiously beautiful cardiac neural crest cells, if I may say so myself. So this cross-section here is just looking at, again, I'll just try to stabilize here, and I know it's really bad, E11.25 developing, so you have your heart down here, right ventricle, left ventricle, here's your spinal cord back here. So we're looking at qualitative evaluation of the loss of cilia or presence of cilia here. So on the right, we have these images showing you, again, how small these cilia are. In our control animals, we're seeing in the pharyngeal area the presence of primary cilia, both in TD tomato positive, so neural crest cells, as well as coming from non-neural crest derivatives. In the mutant at E11.25, this is where we're seeing the loss of cilia. We're beginning to see it around E9.25, and by E11.25, this is when we see the pronounced loss. So the pharyngeal arch is really just our sort of reference region, but the area that we're most interested in is the outflow tract of the heart, which becomes the great vessels as well as the ventricles. So the outflow tract here, we're showing you that in the control animal, we're still seeing cilia intact in the TD tomato positive mutant cells, but in the outflow tract of the mutant, we're not seeing any cilia really. And obviously, this is a reference image, but this is the qualitative picture or piece to the puzzle. When we quantify that, so here's the quantitative data for the early time point at E9.25. That's when we're really beginning to see the loss of cilia from these cardiac neural crest cells. So that's in terms of thinking about mechanism, how we're trying to target which players are the most important in terms of signaling mechanisms and transduction. So we're beginning to see that statistically significant loss at E9.25, and then by E11.25, that's when we're starting to see these drastic changes, not only in the pharyngeal area, but in the outflow tract, as well as, again, this ventricular myocardium. So dogma aside, we are seeing neural crest contribution to the developing ventricular myocardium. That is something that we can talk about. I'm happy to answer questions on, but more importantly, we are also documenting that these neural crest cells are also losing their cilia and is in fact statistically significant. So the resulting phenotype here, so take away the cilium from these cardiac neural crest cells, and what you get is a really interesting phenotype. You get, in addition to outflow tract defects, so here we're showing you one instance of a double outlet right ventricle. So you can see there's a single outflow tract that forms on the right side of the heart. So in addition to the outflow tract defects, we're seeing a very obvious disruption to the integrity of the ventricular portion of the myocardium, which is meant to be the strong, dense muscular portion of the heart that's responsible for contracting and pushing the blood outside of the heart. And that was the piece that really stood out to me. My clinical background, again, I don't have my doctorate yet, but my clinical background is actually as an exercise physiologist. So I was really fascinated by the muscular piece of this phenotype. And so what we did was we started quantifying what was going on, or what we thought was going on here. And actually, we've been able to label the phenotype as a non-compaction cardiomyopathy. And clinically, diagnostically, that is typically evaluated by measuring the thickness of this trabecular or spongy myocardium as it relates to the thickness of the compact or dense muscular portion of the heart. And again, diagnostically, that's considered to be significant at a ratio greater than 2:1 in humans. So in terms of quantifying the trabecular thickness, these data are just showing you that we are, in fact, seeing a statistically significant increase in trabecular thickness with the loss of primary cilia in cardiac neural crest cells. And again, that ratio, in particular in the left ventricle, is also statistically significant. The important piece to emphasize about this slide is that, in fact, we're seeing this phenotype persist past the embryonic time point. So these mutants are living one day past parturition, and the non-compaction phenotype is persisting to the postnatal period. So we were lucky enough to have the opportunity to try to assess this, and this sort of methodology is something that's in the works in our lab. We've developed a really nice model for non-invasive perinatal ECG in baby mice. And obviously we can't anesthetize them because they're sort of on the brink of death. So we've had to really improvise here. Anyways, so this is here where we're talking about more or less preliminary data, but the take-home message is that there is an electrophysiological aspect to this phenotype. And specifically, we're seeing incidence of bradycardia in all of the mutants that are lacking cilia of their cardiac neural crest cells. And there's something going on with the propagation of that signal. So when I say absent P wave, really what we're saying is, I had a medical student ask me about this, and they were too afraid to ask a question like, if they don't have P waves, wouldn't they be dead? Absolutely. But so they're not really absent. What's happening is they're hiding in this very large amplitude of a QRS complex. So something's going on with the rhythm and the rate of transduction through the electrophysiological aspect. Okay. So the last phase of the experiment was really to look at the signaling aspect, and we did so by introducing a mutant smoothened receptor. Again, that's the receptor that will be in the mutant context is going to be operating in the absence of sonic hedgehog. So we combined that with our cilia conditional knockout, and what we found is a very similar phenotype. In fact, so here's our control. Here's our original conditional knockout, and the mice that had the mutant smoothened receptor in addition to some intact cilia actually had the most pronounced or degraded cardiac phenotype. In comparison, or ironically, what we were seeing is that the mutants that were losing their cilia, so the homozygous mutants rather, we're actually seeing almost, I don't want to call it a rescue yet, but we're seeing some maintenance of that compact portion of the myocardium. And whether that's hypoplastic at this point is something that we have yet to determine and will need to do so. But when we quantify that again, what we're seeing is exactly what we're seeing in the qualitative aspect. In fact, we're seeing a very similar phenotype in terms of the hypertrabeculation of the mutants, but in the mice that have the cilia conditional knockout, combined with the mutant smoothened receptor, they're maintaining some of their compact thickness. And another thing to point out here would just be that we're also seeing an enormous amount of variability, and that's something that we need to address. And obviously the numbers here are small. From a mouse work standpoint, this Cre driver has to be, we can only use Cre positive females. So from a technical perspective, it just takes a long time to make all of these mice. So in terms of just, again, coming back to this idea of non-compaction cardiomyopathy, we are seeing, again, a persistence of this non-compaction phenotype. And in the mutants that are having the smoothened receptor with some intact cilia, they are, while they have the highest thickness of trabecular myocardium, they also seem to, the mutants lacking cilia seem to keep some of that compact portion of the myocardium intact. And we haven't yet assessed whether they have, whether perinatal lethality occurs. It likely does, and if we can do so, we would proceed to collecting some of the electrophysiological data there. But the next step of the question is really, when will Lindsey finish her PhD? So I gotta focus. I've got to stick to my exploring the mechanism as much as I'm so excited by all of these other pieces. So starting with the endocardium, so we know that maturation of the ventricular portion, proper maturation rather, is really dependent in many ways upon the endocardial layer. So what we're seeing with the elimination of primary cilia from cardiac neural crest cells is that when the cilia loss begins at around E9.25, you're seeing an obvious, what we're calling disorganization of the endocardium, highlighted here in green, by E10.25 or by 10.5, which is developmentally, that's a big window, but the qualitative differences are pretty obvious. So what do I mean when I say disorganized? There's a very clear sort of continuous level of signal here in your control. It's very organized and ribbon-like, whereas in the mutant, you have this very discontinuous signal that sort of looks like a bomb went off everywhere. And that's, again, qualitatively quite interesting. So what do we want to take away from all of this? I think there are two big aspects to this project. In terms of thinking about cilia in cardiac neural crest cells, I think we've been able to answer this question that, in fact, cilia of cardiac neural crest cells do contribute to the maturation and the formation of ventricular myocardium. Additionally, loss of cilia are clearly implicated in formation of many of these congenital heart defects, including but not limited to outflow tract defects, ventricular septal defects, and impaired ventricular maturation, specifically the compaction process. And finally, primary cilia of cardiac neural crest cells appear to be essential to this part of heart development, both in the embryonic development phase as well as the early postnatal period. And then the other piece of the project is really related to the signaling and mechanistic aspects. And this is the piece of the project that we really haven't comprehensively answered yet. We're still working on it. But I think at this point we can say that forced or constitutively active hedgehog signaling via introduction of this smoothened receptor perturbs ventricular maturation. There's an obvious connection with the genotype and the phenotype. Also, we have concluded that with this mutant hedgehog signaling, that's going to lead to the most severe of the phenotypes. But we're still coming back to this idea that is this mechanism, the loss of primary cilia in cardiac neural crest cells, is this carried out specifically by hedgehog signaling? And how is hedgehog signaling specifically impacted? Is it upregulated or downregulated? What exactly is going on? So coming back to, given the timing of the loss of the cilia, what are our relevant signaling pathways to consider? Aside from hedgehog, and specifically sonic hedgehog, we have the obvious candidates, which are notch and neuregulin, which in the endocardium are well documented, and in particular by our colleague Doug Sawyer over here, in terms of being really crucial regulators in this compaction process, both in the signaling piece as well as the mechanical aspects. Additionally, we have that the cardiac neural crest cells are going to contribute to the ventricular myocardium, but again, what are these mechanistic considerations? And I think, at this point we can say that the hedgehog signaling piece, we're looking at what we're calling in our lab as a sort of a Goldilocks scenario, where too little hedgehog signaling is not going to give you a properly developed heart. Too much hedgehog signaling is going to give you a very, very poorly developed heart. So we're ideally needing something right in the middle. There's an ideal amount of hedgehog that is required for both outflow tract as well as ventricular development in the healthy heart. So with that, I'd just like to thank my PhD committee members. I'd like to thank you all for having me here today, and for all of the sponsors. And it's a true honor to be here. I really greatly appreciate the opportunity. And thank you to all of the collaborators and the medical students who have helped me and worked tirelessly collecting ECGs at 2 in the morning. It's a character builder though, right? Yes. Yes, they're putting their time in. So with that, I guess I can take questions.
Lucy Liao Research Institute. Lindsey, nice work. I'm curious about your IFT88 heterozygous mutants. Do they just have fewer normal cilia, or do they have abnormal cilia?
So in terms of how I can definitively answer that question, the question was what happens to the IFT88 homozygous mutants and their cilia? How much do they lose, and are the cilia that are intact still morphologically maintained? Is that right?
I'm asking about the heterozygous mutants.
Oh, the heterozygous. I'm sorry. Yes. So yes, I'm glad that you noticed that. So of the heterozygous animals, we haven't definitively quantified how many cilia, the percentage of cilia that are intact. It seems to be around 30%. The problem is that cilia are only expressed during certain aspects of the cell cycle. So as soon as the cell is beginning to divide, which happens rapidly during development, the cilium disappears. So we can only get these sort of snapshot pictures of that. That being said, so around 30% are being intact. And from a phenotypic standpoint, I didn't talk about this, but it was clear from the data, we may also have a heterozygous phenotype, specifically, specifically as it relates to the non-compaction piece. So the heterozygous animals do not have outflow tract defects, but they clearly have statistically significant non-compaction of their ventricular myocardium. So in terms of what comes next, we have another medical student that's going to be collecting ECG data on those adult mice this summer. And we'd like to do some assessment of the muscular aspects of those hearts. And then also in terms of thinking about signaling, we've implemented an additional control mouse. So we've introduced the Cree and the tomato into a wild type background to get rid of this IFT88 complex so that we can compare even the heterozygous phenotypes to these TD tomato, what we're calling TD tomato controls that don't have that IFT88, and then statistically comparing the presence or absence of cilia to that as well to confirm that. Yeah.
Very nice talk. The statement that cardiac neural crest cells colonize the ventricular myocardium requires you to absolutely believe that the Wnt1 Cree driver is only expressed in cardiac neural crest cells. Mm-hmm. And sometimes that's not true for some of these genetic reagents. Are there any cardiac neural crests Cree drivers for other genes that you might be able to test, or another graduate student might be able to?
Well, yeah, so yes, the answer is yes. And again, given the time limitations to this talk, I didn't really go into that. But to date, we've used two different neural crest-specific Cree drivers, the Wnt1 Cree1 coming out of the McMahon lab, as well as the Wnt1 Cree2 coming out of the Soriano lab. And those are both Wnt Cree drivers, so that doesn't answer your question. But we have explored this, and the answer is yes, there are multiple other models out there, one of which we have tried, the P0 Cree, which is a neural crest-specific Cree driver. We were not able to successfully see any tomato signal in the cardiac region using that mouse. So we are still a work in progress on that. Ideally, yes, we do want to show this in an additional neural crest-specific Cree driver. But I will say that we have used an additional primary cilia conditional knockout, specifically targeting the KIF3A background, which is another cilia-specific gene. So even though both of our Cree drivers are Wnt1 Crees, we've shown a similar and quantified this statistically significant phenotype in both an IFT88 conditional knockout as well as a KIF3A conditional knockout. So that phenotype has remained the same and has remained statistically significant. So for what that's worth.
So that was a terrific talk, and I want to congratulate you on including so many medical students in your research project. So hopefully
they're great. There's one actually in the back now.
Terrific. Hopefully that'll inspire medical students to pursue translational science, which is a very rewarding part of many careers. I have a question about the human correlate to IFT88 and smoothin receptors. And do you know if there have been any mutations localized to either of those genes that correspond with LV non-compaction or outflow tract abnormalities?
Okay, so this is where my experience maybe ends. What I can say definitively is that so the biggest issue at this point in terms of the translational aspect of this project is that non-compaction, at least in the cardiac community, seems to be sort of divided amongst people that think it's clinically significant and people that don't. And also, is it considered to be sort of a comorbidity or a co-occurring symptom as opposed to its own cardiomyopathy? So that piece is difficult because it means that in the human populations, we don't have these codes in the clinical data to really effectively and comprehensively quantify that. So we're working on that now in terms of just trying to compile what has been coded, even if it's qualitative notations of excessive trabeculation. And so we're trying to address that. But in terms of human mutations related to IFT88, I can only speak to that these mutations associated with primary cilia, i.e. this class of diseases called ciliopathies, is mostly studied in the kidney. So there's a lot of renal conditions as well as co-occurring cardiac, what are like symptoms, I guess. So the field is slowly evolving. I don't know if Dr. Tucker would like to speak to this. He is the PI on the project specific to IFT88 mutations in humans. Are you aware of any of those?
No. So the problem with knocking out IFT88 in humans is that it tends to result in... Thank you. So IFT88 is essential for the construction of all primary cilia. And the problem with primary cilia, and the advantage of them, is that they're essential for the development of practically every organ system in the body. And so if you perform similar types of mutations in humans, you simply don't get humans who are compatible with birth. So it's a very difficult thing to look into. So as far as I'm aware, there are no reported mutations in either KIF3A or IFT88 that result in non-compaction phenotypes. So as such, I think potentially the heterozygous model that we're using could be a better system for looking into that. But in the end, the answer is no. The answer is no. There are not any reported IFT88 mutations that lead to this particular type of condition. That being said, there are also none that are leading to other, better known types of congenital heart defects, like ventricular septal defects. And I think the answer again lies in the fact that we simply don't recover these embryos.
Yeah, and I think one final thing I would mention is also that so we do have the sequence for the mouse IFT88 gene, but we do not have it in the human. So this is something that we've talked about with our collaborator, Dr. Morin, potentially in some of these repairs for ventricular septal defects, some of the discarded tissue, maybe there's an opportunity for sequencing there. But where we don't have a reference sequence yet, that's, unfortunately a little bit further in the future. Yes?
For a point of clarity, as an aging internist, I'm fascinated to learn that there's such thing as a primary cilium. I mean, this is cell biology at its best. So
isn't it though? Yeah.
But just from a point of clarity, you speak of loss of primary cilia, and you're actually it's not elaborated in the first place, presumably in the cells that are deficient in have genetic gene. Is that right? You speak of it as being evanescent over time, but I'm assuming that they're not losing them. They're just basically it's a cell organelle that's not developed. Or do you know why the cells do not have the primary cilia that have this genetic defect?
Yes and no. So believe it or not, the primary cilia field is small, although expanding. It's a field that is highly specialized. So I refer to the cilium as disappearing because there are many different mechanisms by which we can look at modulation of the cilium. So whether it's from a molecular biology standpoint, whether it's absorbed or resorbed, these are all very specific. As a cell biologist, I'm sure you understand that we have to be careful about how we describe this phenomenon, but we don't actually. What we do know is that with this particular IFT88 background, the IFT88 gene that is transcribed still produces an IFT88 protein, but it's a non-functional protein. So some of the cilium is intact, but it's nowhere near the length that we're seeing in the non-mutant cells. That being said, and again, back to Dr. Liao's question, we absolutely need to further characterize the morphological aspects of the cilia that we are seeing. So in other words, with an organelle that's very sensitive as the cilium is, changes in orientation, changes in length, especially where they may serve a mechanosensory role, that absolutely needs to be described and quantified. If there are changes in the length of the cilium, what does that change look like, and is it statistically significant? And furthermore, what's happening upstream and downstream to that as well? Yeah.